Hello. Today we are starting Chapter 3 Cells, the Living Unit. This is Part A. So, cell theory is basically stating that the cell is the smallest structural and functional living unit of the body. So, our organismal functions depend on the individual and collective cell functions. And the biochemical activities of those cells are directed by their specific subcellular structures. So their mitochondria, their Golgi apparatus, their rough ER and smooth ER. All of that depends on what they're doing. And life continuation has a cellular basis. So, there are over 200 different types of human cells. They differ in size, shape, subcellular components, and function. My favorite statement is structure dictates function, and that is definitely true for cells. So, their structure, how they look, is going to dictate how they act, and vice versa. So we have erythrocytes or red blood cells, and you can see that they're circular with a divot in the middle, and that divot is to carry oxygen molecules throughout the body. We've got fibroblasts, which are long and spindly, holding things together, epithelial cells that connect body parts and linings. We've got skeletal muscle and smooth muscle, and these move organs and body parts. We've got fat cells for storing nutrients, macrophages for fighting diseases, sperm cells for reproduction, and nerve cells for gathering information and controlling body functions. And you can see that they all look different because they all have different functions. So a cell has some generalized structures in common. So all human cells have three parts: a plasma membrane, a cytoplasm, and a nucleus. There are only two cells that don't have a nucleus, and we'll get to them later. Plasma cells have a flexible outer boundary that keeps the cell together. The cytoplasm is the intercellular fluid containing organelles, and then the nucleus is the control center that houses the DNA. So we can see all of the parts here of a basic animal cell. We can see right around here is that plasma membrane. Here's our nucleus with our nuclear envelope and our nuclear pores, our chromatids and our nucleolus. Here we can see our smooth endoplasmic reticulum and our rough endoplasmic reticulum. We've got a Golgi apparatus right here. We have some vesicles with some peroxisomes in them. Our mitochondria are microtubules, so we can see microtubules here, intermediate filaments a little bit bigger down here, centrioles right there that help with dividing cells, some lysosomes, which are these little red things, cytosol, which is the jelly-like fluid, where centrosome matrix, which is our microtubule cytoskeleton elements here. So the first thing we're going to talk about is the plasma membrane, and that is a bimolecular layer of lipids and proteins in a constantly changing fluid mosaic. So they like to brush past each other and move, kind of making this fluid model. It's almost like a meshwork. Plays a dynamic role in cellular activity, so it helps bring cell things in and out of the cell. So it also helps separate intercellular fluid from extracellular fluid. So it keeps the inside stuff in, the outside stuff out. And intracellular fluid is more important than extracellular fluid that surrounds the cell. So they want to keep everything in balance, but it's more important to keep things that need to be inside the cell, things that need to be outside the cell outside the cell. So the plasma membrane is a super important function, or has a super important function in that regard. So we can see it here. It's a phospholipid bilayer, which you should all remember from biology. So we can see those phospholipid polar heads here, and those non-polar tails here. These guys are hydrophilic, while the tails are hydrophobic. So they keep that, what is that called? Fluids where they need to be. So we have an outward facing layer and an inward facing layer. You can see our cytoplasm here and our extracellular watery environment out here. In that wall, we also can see glycolipids, glycoproteins, cholesterol. And if you remember, cholesterol right here is a steroid. Integral proteins, attachments to the cytoskeleton, peripheral proteins, um, and then glycolics out here that's sticking out of the cell. So that plasma membrane serves more than just a barrier. It also tells things, it can come in or it can come out. It regulates the the chemical electrogradient of a cell. It also talks to other cells, so it sends signals out to other cells. And we're going to talk all about this a little bit later in the slides. So, membrane lipids, about 75% of that membrane is going to be those phospholipid bilayer, so the phospholipid, with the phosphate head, which is polar and hydrophilic, so it likes that water, and it unequally shares those, right? Yes. And then the fatty acid tail, which is nonpolar and hydrophobic, which does not like water. So if you remember from the last chapter, we talked about the different bonds and the lipids. This is why they're important. Then we have glycolipids. About 5% of those are glycolipids, which are polar sugars that group on the outer membrane surface. So they were those green things right here, right? Outer surface of the membrane. And then about 20% of it is cholesterol, which increases membrane stability and fluidity. So it helps keep that membrane together and keep it fluid. So these are our steroids right here. Cholesterol. Lipid rifts. About 20% of the outer membrane is a lipid rift, and they can contain phospholipids, sphingolipids, and cholesterol. And many function as stable platforms for cell-to-cell cell signaling molecules. So those rafts are just areas where it's a lot of different lipids to help with that signaling. We also have membrane proteins. So integral proteins are firmly inserted into the membrane. Most are transmembrane membranes, so they go from one side of the membrane to the other, and typically they function as transport proteins, enzymes, or receptors for cell signaling. So they're either going to let things come in or out, make reactions happen faster, or take in information and tell the cell what to do. We also have peripheral proteins, just like peripheral vision. They're loosely attached to an integral protein. They include filaments on the intracellular surface and glycoproteins on the extracellular surface. They can either be enzymes, motor proteins, cell-to-cell links, which we'll talk about a little bit later, provide support to an intercellular surface, and form part of the glycolics. So they either speed up reactions, move the cell around, connect either via tight gaps or loose or gap junctions or tight junctions. They provide support, so they provide anchors for the microfilaments, intercellular matrix, and they can also form parts of the glycolyx, which is the outside of that cell. So here we can see again, right? Integral protein all the way through. A peripheral protein that's being linked to fragments or filament of the cytoskeleton, right? Out here, another peripheral protein on the outside. We don't exactly know what that's doing. Here we've got a channel protein. So you can see there's different functions to the different shapes of proteins. So, functions of membrane proteins, we either have transport proteins, receptors for signal transduction, attachment to the cytoskeleton, or extracellular matrix. And we're going to talk about each one of these individually. So, transport is a protein that spans the entire membrane and may provide a hydrophilic channel across the membrane that is selective for a particular solute. So, some transport proteins use ATP as a source of energy to actively pump substances across that membrane. Some do it passively. So some require ATP and some don't. So we have active transport and passive transport. We also have receptors for signal transduction. So this is a membrane protein that is exposed to the outside of the cell and may have a binding site that has a specific shape that fits a chemical messenger, such as a hormone or even antibodies have specific shapes. So the external signaling then may cause a shape change in that protein. So if you change the shape, you change the function. That initiates a chain of chemical reactions within the cell to produce what the body needs at that particular moment. We also have attachments to cytoskeleton and extracellular matrices. So elements of the cytoskeleton, which is kind of like the bone system of the cell, or the extracellular matrices outside the cell may anchor to these membrane proteins, which help maintain these cell shapes and fix the location of a certain protein to a spot and help fix that cell to that spot as well. Others play roles in cell movement or binding adjacent cells together. So you see this a lot in tight junctions and gap junctions with that extracellular matrix links, especially for cell communication. You want those good junctions there so your skin isn't moving around. That would be creepy. We also see enzymatic activity, intercellular joining, and cell-to-cell recognition with these membrane proteins. So enzymatic activity is a protein that is built into the membrane that may be an enzyme with an activation site. So when it's exposed to a substance of an adjacent solution, in some cases they can act as a team, but if they're exposed to a substance, they then help bind it together and produce another substance. We see this a lot in the metabolic pathway, like this ATP cycle, the cyclic AMP cycle. To produce ATP, we need those enzymes to make things hug and combine them to produce the products we need. We can see a metabolic pathway here. So the little green thing goes into the enzyme, it changes its shape to the triangle then goes to a different enzyme to make the square. Right. We all know enzymes increase the chemical reactivity of a substance. We also have cellular joining. So these are membrane proteins that connect to adjacent cells by hooking together in various forms of intercellular junction. Some membrane proteins called CAM of this group provide temporary binding sites that guide cell migration to cell interaction and other cell to cell interactions. Some are permanent, and that's where you get those tight junctions and those gap junctions, and that interlocking with that extracellular matrix as well. We also have cell to cell recognition, where they have those glycoproteins on the outside that serve as identification tags that are specifically recognized by other cells so that that cell doesn't get eaten by a macrophage, it recognizes self, and B, so that we can curve cell overgrowth. And these cell to cell recognitions help with curving neoplasia, which is cancer eventually. So we have three types of junctions that I keep referencing two of them. So we have tight junctions, Demonzones, and gap junctions. So tight junctions prevent fluid and most molecules from moving between the cells. And these might be, where would you think these might be useful in the body? Skin. Skin is where they are most useful in the body, right? We don't want things getting through fluids, most molecules. We don't want that getting through our skin. We want to be able to control what goes in. So you can see a tight junction here is an impermeable junction that prevents molecules from passing through. You got the basement membrane here, and then these plasma cell adjacent cells here with microvilli on the top, but there is very little intercellular space. It is almost non-existent. You can see these junction proteins here, and those gaps are those intercellular spaces. So they want to be very close together, snug and tight, so nothing can really get in. Now we have Demonzones, which are rivets or spot welds that anchor cells together. Where do you think these might be useful in the body? So you can see them here. The spot welds that are held together. These are more useful in things like intestinal lining, blood vessels. We want things to be able to go through, but not super a lot. So they bind adjacent cells together. They help internal tension, reducing the network fibers. It's typically keratin. They're linked together by glycoproteins above a basement membrane. Intestines is another good example. And then there are gap junctions. This is where transport membrane proteins form pores that allow small molecules to pass from cell to cell. For the spread of iron, this is typically used for the spread of ions between cardiac and smooth muscle cells. We see this a lot. We also see this a lot in nerve cells. We want that free moving electrogradient to come in and help contract or extend our muscles or tell us to do something, make us breathe. So we can see them here, right? It is literally just two channel proteins holding hands, letting things move from one place to another. We see this a lot with nerve cells as well. So finally, plasma membranes are selectively permeable. So some molecules can pass easily through while others cannot, and it just depends on what that molecule is. Big chunky molecules aren't going to be able to move through that small space. Small molecules can. So Membrane transport, we have our passive transport, which no cellular energy is required. Substance moves down the concentration gradient. There's no active doing of anything, it just happens. Active processes is where you need ATP to do something, and it occurs only in living membranes. Dead cells can take things in without energy all the time. Living cells can't. Only living cells can use ATP. So, the passive process, what determines whether or not a substance can be passively permeable, um, is going to be the lipid solubility of the substance, the channels of appropriate size, or a carrier protein. So, we have a couple of different um, proteins that we can use. We've got simple diffusion, carrier mediated facilitated diffusion, channel mediated facilitated diffusion, and osmosis. So simple diffusion is just going straight through that phospholipid bilayer. These things are non-polar lipid soluble hydrophilic substances that go straight through the phospholipid bilayer. They need no help. They can just walk through. They don't need anyone to hold their hand. They are just strictly walking through that membrane. Right? Lipid soluble substances on the outside can just go straight through. No problem there. Facilitative diffusion contain lipophobic molecules like glucose, amino acids, and ions, which need a carrier protein or a channel, both in which exhibit specific selectivity, are saturable, so the rate is determined by the number of carriers or channels on the cell membrane, and can be regulated in terms of activity and quantity. So they can add more, or they can decrease the number, or they can turn things on and turn things off when we don't need anything more in the cell. Oops. Oh, I thought there was a photo. I'm sorry. So, transmembrane integral proteins that transport specific polar molecules like sugars and amino acids are going to be our carrier proteins. They bind the binding of the substrate causes a shape change to let it in. It's like something has to open a door. So it opens a door and it closes behind them. Is how facilitated diffusion with carrier proteins happen. So you see something comes in, sugar comes in, and then the door opens and it closes behind them. So they are carried across the membrane. Now we have channel proteins, and these are two forms. We have an aqueous channel formed by a transmembrane protein and selectively transports either ions or water. We have two types. We have leakage channels, which are always open. Then we have gated channels, which are controlled by a chemical or an electrical signal. So you can see here a channel-mediated facilitated diffusion. This is just a straight hole. Goes straight through. This is mostly ions, selected based on size and charge. We can see this with water molecules and osmosis, but it's just a walkway straight through. Like I said, osmosis is also the movement of water across the membrane. Water diffuses through the plasma membranes. It can either go through the lipid bilayer, or it could go through water channels called aquaporons, which you can see here. It can go through the plasma membrane, or it can go through a channel protein called an aquaporin that helps diffuse water throughout the cell. So the water concentration is determined by the solute concentration, because solute particles displace water molecules. So it has to be, right, depending on the solute the solute concentration. So osmolarity is the measure of total concentration of a solute particle. So when a solution of different osmolarity are separated by a membrane, osmosis occurs until equilibrium is reached. So we either have an isotonic, a hypertonic, or a hypotonic solution. You can see here a membrane permeable to both types of solutions of water. So we have a solution with low osmolarity or low solute content with a solution of greater osmolarity. It's going to equal out. So both solutions will eventually have the same osmolarity, but the volume stays unchanged. Those solutes just move across the concentration gradient. You can see here, once again, lower on the left, higher on the right. But if it is impermeable to solution, both solutions have identical osmolarity, but the volume of solution is greater. So this one, the solutes were able to pass through that layer, while this one, they aren't able to, so you're going to get a volume change. When osmosis occurs, water either enters or leaves the cell, and it changes the cell volume, therefore changing the shape, disrupting the cellular function. Four minutes, and I can finish this up. Tonicity is the ability for a solution to cause a cell to shrink or swell, and we have different types of solution. We have isotonic, which is the same solute concentration as the cytosol. Hypertonic, which is a greater solute concentration than the cytosol. And then hypotonic is solution having lesser solute concentration than the cytosol. So hypo gets bigger, hyper is hyperactive, so it gets smaller, and isotonic stays the same. So isotonic stays the same, hyper is hyperactive, so it shrinks down, and hypotonic gets nice, round, big, fat because it has an O in it. So cells retain their normal shape, size, and function in an isotonic solution, while cells lose water by osmolarity and shrink because of a hypotonic solution. And then hypotonic is water, cells take on water by osmosis until they have become bloated and burst or lyse because they have swelled so much. So, summary of all of our processes: we have simple diffusion, which uses kinetic energy. An example is the movement of O2 through the phospholipid bilayer. Facilitated diffusion also uses kinetic energy, and that is a movement of glucose into the cell. And then osmosis is kinetic energy, which is movement of H2O through the phospholipid bilayer, or aquaporons. I'm going to end here. We will pick up at part B on Wednesday. I will see you then.